And my biology role model is Nina Tandon. So, Nina grew up in a family of engineers. Her parents and grandparents were engineers, and she and her siblings all went on to become engineers. Her parents really focused on STEM while she was growing up. She went to Cooper Union and got a degree in electrical engineering, and later she decided to get an MBA at Columbia. She did this because her brother was diagnosed with genetic retinal disease, and she believed that the study of genes might be the way to treat that. At around the same time, the Human Genome Project had just wrapped up, and she was inspired by it. And later, she started a biotech company called Epibone, which has recently received FDA approval for clinical trials in the knees. Nina's hobbies. As a child, she really enjoyed community theater and sewing, and she made a big effort to keep the humanities in her life as she grew up. As an adult, she enjoys metalworking, running marathons, and yoga. Why I admired Nina. First of all, I admire her approach to continuing her education and not being satisfied with just what she's learned. She continues to learn more and, research and study more. And I really like that mindset. And in addition, the research she's doing could really help people like my father, who has serious knee problems and he's had them since college. And the research she's doing could really change his life. Her focus. So Nina was an electrical engineer and she later got an MBA. So she took what she learned as an electrical engineer and kind of applied that later on to medicine and biology. Specifically, she focuses on stem cells. And the research she's doing with her company Epibone focuses on something called engineered allogeneic osteochondral grafts, which are basically focusing on cartilage, regrowing cartilage, specifically in the knee. Okay, so this is one of Nina's recent papers. It's titled Alternative Direct Stem Cell Derivatives Defined by Stem Cell Location and Graded WNT Signaling. So this was from 2022, and this study focuses on stem cells, cells that can differentiate into a wide variety of cells and their organization, and kind of it's trying to figure out how they work. Specifically, it focuses on Drosophila ovarian follicle stem cells, or FSCs. This study uses a wide variety of techniques, including multicolor lineage analysis, immunohistochemistry, live imaging, and more. But this figure 1, uses a few things in particular. It's about the location of FSCs in a geranium. So that's ovarian follicle stem cells in a germanium, which is the part of an ovary that produces eggs. Parts A and B are schematics of a germanium showing its parts, including the locations of FSCs. C, D, F, G, H, and J use multicolor image analysis to locate these FSCs throughout the germanium by looking at different layers, and they track the different FSC lineages to try and follow the differentiation. Graphs J and K summarize these results and look at the different lineages and their locations. So overall, Nina and her team use this research to kind of extend and revise their understanding of FSC behavior. They found that germaniums contain between 14 and 16 FSCs organized into 3 layers: And the specific location of these cells within the structure can determine how they differentiate. Next steps. So far, Nina and her team at EpiBone have really done a lot. In July 2023, they received IND, investigational new drug approval from the FDA to begin testing lab-grown knee cartilage in humans, which could help people with damage from sports injuries, trauma, and more. In the future, her next steps are making the technology simpler so it can be done on a larger scale while still making personalized tissue. And these are my citations. Thank you so much for listening.